So as important as these fibrous proteins are, right, they represent a relatively small class in terms of the diversity of protein structures um, compared to this next family of proteins we're going to talk about, which are, of course, the globular proteins, right? The globular proteins are the bulk of what we're going to think about in this course, and these are structurally much more complex than the fibrous proteins. The structure of globular proteins is still very much specified um, by the particular amino acid sequence that makes up that protein. But it's not governed by simple repeat patterns in the way that most fibrous proteins are. So it's a little harder to see, and it's been a long-standing problem since at least the 1940s or 1950s in understanding how that information propagates from amino acid sequence to three-dimensional structure. And it's a problem we still have not completely solved, right? But for globular proteins, one thing that is true is that the structural hierarchy that I've been talking about that propagates from primary sequence up to secondary sequence, up to tertiary sequence, and then up to quaternary sequence is much easier to see. That, as we just saw, that higher structural hierarchy is still there in fibrous proteins, but because in fibrous proteins, their properties are so dominated by their secondary structure, it's harder to see how that maps, right? Their three-dimensional properties are really sort of elaborated elements of secondary structure. In globular proteins, however, that's less true. Globular protein, the structural hierarchy is a lot easier to see. And let me just give you an example of that. So here's a protein we're going to spend a lot of time talking about here really soon, hemoglobin. Right? So here's hemoglobin's primary sequence. That is it. That's the sequence of, I think that's the A subunit. Can't remember which one I pulled. But that's more or less the amino acid sequence, right? Right. And the amazing thing is that amino acid sequence, every time under physiologic conditions, will specify a particular set of secondary structure. And in the case of hemoglobin, that's almost completely alpha helix and turn and loop, right? That's pretty much all it makes. And those alpha helices then get stacked against each other in a very particular three-dimensional arrangement. And then there are two different variants. There's an A subunit and a B subunit, or an alpha and a beta subunit for hemoglobin. And you have two of each that come together in quaternary structure of hemoglobin to form the hemoglobin tetramer. Right. Okay, and you can see here highlighted in green is one subunit and blue is the other, and there's the heterodimer, and then a symmetric heterodimer on the other side forms the hemoglobin heterotetramer, right? Okay, so you can hopefully sort of begins to give you a sense of how that primary sequence propagates into this three-dimensional structure that is an ordered functional protein whose mission in life, as we're going to see, is to bind and release oxygen. Okay, so another important property of globular proteins is the way in which their amino acid components are distributed, right? We've already talked about this a little bit. So globular proteins, less so fibrous proteins, have kind of a clear inside and a clear outside. And for globular proteins, the basic rule of thumb that you hopefully remember is that the interior of the exterior of globular proteins, shown here mostly in blue and this sort of neutral gray color, are the polar and the charged amino acids. And they're pretty much mostly what's on the surface. These more yellow colored guys, which have a little bit of surface exposure, but when you slice down into the protein, it becomes much more obvious. These yellow amino acids are the hydrophobic amino acids, and they pretty much fill up the core of the protein. So globular proteins more or less follow this basic rule of the polar and charged amino acids on the outside, the hydrophobic amino acids are buried on the inside. The other thing you note is how well packed the interior of that hemoglobin monomer happens to be. This is, again, is hemoglobin. Look how well packed those side chains of those hydrophobic amino acids are. That's a really important property of globular proteins, and we're going to discuss it more when we talk about lambda repressor in the context of the paper from Lim and Sauer that we'll discuss here soon. Okay, some other general properties of globular proteins is they tend to be compact They're less frequently extended. They can become a little extended, and you can use a globular protein to make a more extended structure. Things like actin and tubulin are really good examples of that, right? But in general, the individual subunits of fibrous proteins, or excuse me, globular proteins, tend to be significantly more compact. They also are vastly more structurally diverse than fibrous proteins. There are just an enormous number of ways to build a globular protein, from structures that are completely all alpha helical, right? Here's an example of one, myoheme here's another one that's almost completely alpha helical with a little bit of beta sheet, to proteins that are almost completely dominated by beta sheet. This immunoglobulin fold is a completely beta sheet structure, it's so-called beta sandwich. And then you can mix it up, right? Examples of forming a parallel beta sheet with flanking alpha helices, or mixtures in other ways. They're just a huge number of ways in which secondary structural elements can get packed into three-dimensional space to form a globular protein. Now, interestingly enough, right, even though that space would seem vast, right, and it is enormous, it's not infinite. And even within the huge space that's available, biology or evolution seem to have settled on certain structures that it uses over and over and over and over and over again, right? So the space, though not infinite, is vast, and evolution has seemed to mostly operate even within the restricted subset of that space. And so you see what structural biologists call motifs used again and again and again that appear to be particularly useful, or were identified early in molecular evolution and just became so useful they just got elaborated from a core initial structure. So some examples of these are like having a beta sheet with a piece of alpha helix over the top, and then an underlying piece of beta sheet. These two guys would be forming, the two pieces of beta sheet would be forming those lateral interactions with the alpha helix laying across the top. Beta hairpin, and that's like the classic way in which you build an anti-parallel beta sheet. You don't have to do it that way, but that's a really good way to do it, because you just staple in a beta turn and you've got an anti-parallel beta sheet. There's that alpha helical stacking interaction that's likely in an alpha-alpha stack like this is going to be a coiled coil like we just saw. You can make beta barrels. You take this sort of basic idea and you just wrap it around and close it up, and you form a closed beta barrel, right? Multiple different ways you can do this. And a way that gets used over and over and over again to make a parallel sheet beta barrel is to take a sheet of beta, take a piece of beta sheet, bring it out in a loop, make an alpha helix, loop that back around into another piece of beta sheet that then stack against. And so you get this parallel beta barrel with these buttressing alpha helices around in the exterior, right? So a really important point here is that, unlike with the fibrous proteins, where you got that one kind of structure that was encoded by that simple repeat pattern, a wide variety of different amino acid sequences can be compatible or are compatible with any one of these motifs or any given fold. A huge number of different amino acid sequences can all make the same basic type of structure that, like these that are shown here. But that doesn't mean that all sequences are allowed in any given fold type. Even though the sequence space is huge that allows these different types, it's also not completely infinite. There are lots of sequences that are incompatible with these particular three-dimensional arrangements and wouldn't be allowed, right? Okay. So in addition to certain motifs being used over and over and over again, there's an additional level of sort of structural functional specialization, in that certain types of these folds appear to be really good for doing certain types of things. A good example of that is the way in which this kind of structure, this sort of alpha beta barrel, gets used. And one of the ways it gets used a lot is to make enzymes, right? We haven't talked about this yet, but we're going to. But the central core, there's that parallel beta barrel. Here are those buttressing alpha helices around the outside. That central parallel beta barrel turns out to be a fantastic way to build an enzyme active site, right? To interact with stuff and do chemistry, right? This is also sometimes known as a TIM barrel for the enzyme triose phosphate isomerase that we're going to see again before long. Okay, there's other examples, right? So you want to bind calcium, right? Turns out there's a fairly simple way to build a calcium binding loop with a couple of pieces of alpha helix and a simple loop turn, right? This is known as a so-called EF hand, right? Where this one helix is sort of like the finger of a hand. The loop is kind of like another finger, and there's another piece of helix down here that's the thumb. This, if you use the right amino acids and do it in the right way, and they can be quite structurally diverse, this is a great way to bind a calcium atom, right? And of course, one of the famous proteins that use that particular structure is the so-called calmodulin, right? Which is super important protein, calcium signaling, right? Okay, so another example, and I could go on and on and on, but just one more example. The so-called zinc finger. Many of you, I'm sure, have heard of these in your studies in molecular biology. Zinc fingers are used in lots and lots of DNA and RNA binding proteins and transcription factors and other things to present a face of this alpha helix for interacting with nucleic acids. And it turns out a really good way to build a stable and small piece of secondary structure that makes this helical piece nice and stable is to sort of staple the back side of it with a zinc atom connected to a simple two-stranded anti-parallel piece of beta sheet, right? And you hold these two pieces together, stapled together with a zinc atom, a couple of histidines in this case, and a couple of cysteines. So the zinc finger motif gets used over and over and over and over again, both for doing DNA binding and for all kinds of other structural roles. In some cases, not completely clear what they're doing, but this sort of motif crops up again and again and again. Okay, finally, there's a concept. You start thinking about three-dimensional structures of globular proteins that you also need to understand. That's sort of a subcategorization, in a way, of the tertiary structure of protein. And this is the so-called idea of a domain. All right, so here I'm showing you something that's known as the Rossmann fold. It was named, we'll come back to it, by a famous structural biologist who passed away not long ago at Purdue, named Michael Rossmann. And he was the first person to recognize really this idea of structural motifs. And the very first one he identified is this one. It's an NAD binding motif. And he got his name, the Rossmann fold. The idea of every structural biologist, like planting a flag on the moon and naming their own fold, got nixed right after this because it would have been completely out of control. So nobody else gets to put their name on protein folds, but Michael got there first, so he got to plant the flag. So in addition to this motif getting used over and over and over and over and over again to recognize certain types of nucleotides, specifically NAD, where it's used a lot, you'll notice something. It's been the color here is sort of in two different areas here, right? Even though this is a single polypeptide, there are two different segments you can kind of recognize as being kind of two parts. It's like there's the bits of beta sheet, these three pieces of parallel beta sheet, these three helices, and then you have something similar, three more pieces of beta sheet and two helices. And if you sort of thought about maybe if I cut this right there, I can almost recognize those two as autonomous looking pieces that have been put together. This is the concept of a domain, right? It's a little less obvious here, perhaps, in the Rossmann fold. And one of the ways in which it's recognized is sort of like a clear piece of sort of what would almost look like a protein on its own that's connected to another piece by the same piece of polypeptide. In some cases, these domains actually will fold on their own, will structure themselves if they're isolated. Another couple of examples. Here's another one, glyceraldehyde 3-phosphate dehydrogenase. This is an enzyme we're going to be talking about again. And the domain structure is now sort of really clear here, right? Shown here in green, right, is one domain. And then down here in this sort of pink color is the other domain, right? And we can sort of see how these two pieces of this polypeptide are kind of distinct. And then here, finally, here's another protein that's sort of been near and dear to my heart, GRO-EL, chaperonin. In this case, it's even more obvious, right? That GRO-EL has three domains. There's this sort of red domain up here at the top, right? So-called apical domain, and there's this equatorial domain down here, and they're connected by this little green intermediate domain. But the connections here in each case are kind of like just these little bits, right, that hold these guys together. You can almost look at these as three different proteins that have been put together and stitched with some little pieces together. So the idea of domains is sort of a level up from just the simple tertiary structure before you then get the quaternary structure within a single polypeptide. Bigger ones can have different distinctive structural domains that then form the overall tertiary fold of the protein. And finally, then we can take these individual bits of polypeptide and start our folded protein that have their three-dimensional structure, but are not covalently linked to anything else, and start stacking them up. When you start stacking these up, you begin to start talking about the quaternary structure of organized proteins. So in the case of Gruyere, 14 of those guys come together and make this large double-ringed barrel, right? Another good example of this are all the subunits that come together to form viral capsid, right? And the structural diversity here and the function that can get encoded from putting these different individual subunits together to form these larger structures can be enormous and can be quite beautiful, right? In the case of these large viral particles, as well as many of the other large protein complexes that get assembled to do basic biochemistry. And we're going to start talking about those as we go along. Okay, see you next time.